the opponent the question has already reported. So yes, that's all. Thank you.
For your review. So now it is time for questions from the jurors, and you can ask a question to any of the participants of the science. Yes, please.
Thank you for the presentation, and I have a couple questions to reporter. Do you know anything if C11 is toxic for human, about toxicity of C11 for human?
Okay, thank you for your questions. The C11, according to the publications that I read, it doesn't have any particularly harmful toxicity to human, but I think it has some possibility of toxicity that we should also consider about that.
And another, you suggested for future to use protein-based library.
Yes.
Do you have any, why you suggest protein-based library but not sequencing library, like sequencing of infectious microorganisms? Why do you think that protein-based library is better for identification?
Thank you very much. So, of course, we think that the sequence-based library is also very important, but we must remember in the formations of virulency of bacteria or any kind of pathogen, I think the one that the really that with our body is the protein, not the DNA itself. That's why we conclude that probably targeting the protein or making a protein-based library could be a better solution. So, thank you.
Please, any other questions? So, if there are no questions, let's thank every participant on the table, and we can proceed to the grades from jurors. Yes, you can take your places. Yes. So the jurors have to put their grades in their individual protocols, and after some time we're going to announce them. Yes, our first marks. Are everyone ready? Yes, thanks. So, please show your marks for the performance, for the whole performance of the reporter. The marks are 8, 9, 8, 7, 6, 7. Then the marks for the opponent, please. 4, 2, 1, 2, 3, 3. And the marks for the reviewer. 4, 4, 2, 2, 2, 3. Thanks a lot, and it is time for any comments from the jurors, if you have any. Any comments about this science fight? Obviously, participants can discuss some aspects of problems with our experts afterwards during the breaks. So that's all for the stage 1. Now we can proceed to the second stage, where the reporting team now will be the team Watson Screams, and the opponent team now will be the team 5 Samurai. The reviewer will be the team Emotional Synapses. So what is your eternal rejection, Watson Screams? Also number 5, yes? So the team 5 Samurai challenge the team Watson Screams to the problem number 6, which is called Yes, do you accept the challenge? Yes, okay, that's nice. That's nice. You can take your time for preparation. So we are going to discuss problem number 6. Можно.
I will start.
Можно сейчас микрофон можно блокировать. Please announce your name.
I'm Christina, captain of Scream of team Watson Screams. Degree of comfort. RNA temperature of the RNA thermosensor is a temperature-sensitive non-coding RNA that takes part in the regulation of gene expression. The presence of the RNA thermosensor to change the secondary structure of this molecule in response to change of temperature. Objective. Explain the molecular mechanism for triggering a thermosensor. Specify the temperature, then we can use the thermosensor, and offer any possible application for temperature-sensitive RNA. What is RNA? It's a short nucleotide sequences of non-coded messenger RNA. Thermal RNA allocate in 5-stroke non-coded regional upstream of the protein coding gene. Thermal RNA contains the hairpin, which is a small number of mestated nucleotide pair. This mestated nucleotide pair may non-canonical pairs which reduce the stability of the structure. What are RNA thermosensor? The RNA thermosensor can be cis-regulatory thermo-RNA associated with the protein encoding gene, and trans-acting thermo-RNA action from different molecules. Gene is not directly associated with thermo-RNA. problem with the selection of optimal conditional of the thermorNA. In wild type, thermorNA, because it's non-canonical parent, they often cannot predict the secondary structure of the RNA thermometer, effective melting point of the stat, ability of thermorNA to work in off-on mode. The solution of this problem is synthesis the novel RNA thermosensor with the predictional properties. How does RNA thermometer this regulatory? Non-canonic pair in the hairpin are unstable. With the temperature increases, the hairpin structure can melt. In this condition, the ribosome-based SAID or Schneider-Garner sequences can be exposed for binding on the ribosome. Now, ribosome can synthesize the messenger RNA into peptide. If we take thermorNA and target gene, we can get thermoactive regulation gene expression. What can regulate gene RNA thermosensor? The target gene associated with the RNA thermosensor can encode protein inhibitor, fluorescence protein, molecular messenger are not encoding gene and will be small interferon RNA and the aptamers with ribosome activity. They can object the temperature of RNA activity depending on the composition of GC nucleotide pairs. And temperature-sensitive RNA are the target of pathogenicity and virulence. Many pathogenic organisms, bacteria or virus, have pathogenicity and virulence genes associated with thermorNA sensitive temperature of 47 degrees of Celsius. While thermorNA is such organism insensitive to single nucleotide substitution and can become target of gene therapy or super-resistant strains. Also, with the help of single RNA, it is possible to inhibit the pathogenicity and virulence of these organisms. And we will get temperature-sensitive fluorescence as a technique for microscopy study. We can do thermorNA plus on the gene fluorophore. Associated fluorophore can build the different spectrum of the emission. Depending on the temperature, different fluorescence can be expressed, and they will get super accurate intracellular thermosensor. Or improvement fluorescence microscopy techniques, ornament plant, animal. Also, we will improve fluorescent brain ball technology in study of brain and regulate of density and metabolic activity of strains producers. When we sensated the RNA thermosensor with a gene encoding the target protein, will be possible temperature regulate the expression of this protein. And the hedgehog genes containing RNA thermosensor often trigger signaling pathway to stop cell growth in the producer strains. Knockout with gene will be possible to increase cell density. Also, we will take RNA thermosensor plus aptamers. We will get thermoswitch. Aptamers are nucleotides of peptide molecules with specified bind with certain target molecules. The aptamers can be ribosomic activity, inhibit protein binds ligands. Using RNA thermosensor will be create an aptamers inactivity in a certain temperature range. RNA thermosensor associated with small interferencing RNA. Small interferencing RNA is a class of double-stranded RNA molecules operating with RNA interferation. Temperature-dependent inhibition of gene can be carried out with the help of vector RNA thermosensor associated with single interferation RNA. And selective activation of vectors for gene therapy. They take thermodegraded nanocapsule and they locally hit the tumor growth zone, and that leads to degradation of this nanocapsule and activation of the trigger gene. Question hit resistance.
So please proceed to conclusions.
Conclusion. The mechanism of operation of RNA is associated with the conformation rearrangement, and the range of activity thermosensors is quite wide. They make possible different temperature activity of the RNA thermosensor, and they use RNA thermosensor in biotechnology, medicine, research methods, and improvement of existing molecular instruments. Thanks for attention. This is a reference.
So it's time for two clarifying questions, if any, from the opponent. You can ask them from your place. If you don't have any clarifying questions, you can use your 2 minutes for preparation for a position. Yes, please.
Have you mentioned the temperature range of reaction as implied by the presentation? You mentioned the temperature range of action every day. If it wasn't the presentation, that's the question.
The temperature range is quite wide because we can make different temperature activity dependent of GC nucleotide in thermosensor.
Okay, so you also have 1 minute for preparing your please.
Good afternoon, dear colleagues. I represent Team 5 Samurai, and my name is Angela Pavlova.
I'm going to be the opponent for the task about RNA thermosenses. Thank you. Thank Team Watson Scripts for your nice presentation. It covers a lot of very interesting applications, and maybe after this section closes, we can discuss something about them. But I have some questions about the basic theory of RNA thermosenses. The first one, you mentioned that native RNA thermosenses are unpredictable in their structure. There are several structures for them, always, because there are some mismatches in the double-stranded regions. So I think it's why the temperature range is very not so limited to, and it's hard to calculate. So you said that some structures could be some structures could be invented. So we can think of the sequence of RNA to synthesize and then somehow incorporate maybe it in the genome maybe. So my first question is how to predict the properties of the RNA thermosense. So we should incorporate something predictable and something maybe with known properties. So we must assume its structure, its most predictable, its most highly possible structure, for example, and therefore evaluate the melting point of its structures. Also, a lot of your applications that you suggested, they correspond to the applications in vivo, yes, in living cells. So the temperature range is quite a point there, because if you, for example, take neuron cells, so you can't heat them a lot to obtain this change in fluorescence, for example, yes? It will kill them. So you should, I think, consider that. So my main point there is the temperature change, which causes the RNA thermosensor to work, so the melting temperature of the structures. And this point corresponds to the in vivo application. Thank you for your attention.
Thanks a lot. So it is time for polemics between the reporter and the opponent. You can proceed to the polemics.
So you can answer my questions.
You can use the second mic.
Thanks for the presentation. Really useful comments for my presentation, and I know what this temperature range of useful sensor in vivo, and now would be just theoretical approach, because
Okay, everyone knows that a single RNA sequence can form several different structures because of the mismatched pairs and also the incorporation of uncanonical even nucleotides. So it's very hard to predict which one is really the most abundant and the most present one in cells also. So maybe the reporter can suggest the way to do this. the way to do this in soft, in internet, exist special programs for predict properties of the sensor, and this approach really work. In articles, it's it's soft to predict.
So yes, there is a software that calculates the energy, the free energy of the structure. But I wonder if these data, these numbers are really applicable to real systems for cells.
System for cells, just to study and research, but this application in medicine, because it's really little article about this medicine application. Just theoretical approach.
Yes, of course. I think it's connected to the there is no easy way to deliver these RNA molecules to the cell, except for incorporating the genome, and it's not always can be done. It can be done for some maybe cells on the plate or something cultivated, but not for medicine. Can you suggest the temperature region of the action? So you didn't mention any numbers of the range of temperature, so some numbers.
Numbers or I don't know. This sensor exists in several organisms which lives in hot the temperature 20 degrees to 4,000.
I think 4,000 is quite a lot.
400 degrees. The temperature
I think no living organism can maybe
exist with the sensor.
I assume that this method of some regulations can be very not very abundant, but easy to accomplish in extremophile bacteria. But what about maybe some normal cells which live in the region less than 40 degrees? So is there some molecules, RNA, that can be at thermosensor? So for example, they can be useful for some heat shock response, which is stated in the task. So, is there any RNA molecules, and which and what is the melting point for their structures?
The melting point for the structures.
Can it be below 40 degrees, or should it be higher? It could be 40 degrees, yes. If it could, then I think these structures are not very stable. So 40 degrees isn't that much. for a double-stranded region, yes? So what should this, so this region should be really short, yes?
They may make large range nucleotides in this hairpin.
Okay.
And could be many nucleotides were in this structure.
Okay, and maybe you can comment on how to invent a new structure to make it work as a RNA FEMA sensor. So how do we construct it? How should it be thought of making it? Maybe you have some suggestions. I'm interested. How can you?
I need to write some articles about this.
Okay. So,
Because both of you have approximately 30 seconds if you want to add some. No?
no, I think I asked all questions I wanted to ask.
Thanks a lot. So, if the report has nothing to add, then thanks a lot for your polemics. And now we can ask the reviewer to come to the stage to make a review.
Hello, my name is Maria Melnikova from Emotional Synapse, and I'm glad to be here and what I'm doing. Review these great polemics. The information presented by reporter was well structured. It wasn't full, but reporter presented it very well, and thank him for that. In our opinion, there is some missing information about expression or other using ways of use of RNA and its practical applications of supramolecular or molecular mechanisms of it. But the information presented about luminescence was great. The part about non-body existence and activity isn't full, and opponent noted it very well. I think opponent gave us very great polemics, and it was very pleasant to see it. I want to add a little bit about existence of RNA in different gradient of temperature. RNA in extremophiles are survive in rather more big temperatures than in normophiles, but not because RNA is so strong to it and can resist to it, but because extremophils can protect its heart, its RNA from influence of external factors. Thank you for your polemics. It was very pleasant to see it, and that's all.
Thanks a lot. You should stay here. Yes, please, someone from the jurors, any questions? Yes, please.
Yes, everybody. How stable is RNA in organism and in vitro, this RNA thermosensor? I would like to.
So let's start from the reporter. Yes, the question was to every participant, and we should start from the reporter.
How stable is RNA thermosensor in organism and in vitro?
In vitro, understand.
Is there any information about the lifespan of the molecule? How long they live in organism?
The wild type RNA thermosensor are not unstable because this RNA thermosensor don't work in off-on regime, and synthesis artificial RNA thermosensor will be rather stable in vitro and organisms for therapy. Thanks for question.
The same question to the.
Yeah.
Yes, please.
As you know, RNA is not is a fragile compound, so it is it can be subjected to degradation. So there is a problem of maybe incorporating the artificial thermosensors not through the genetical way. So you cannot just apply these thermosensors maybe for cells using not using the genetical incorporation. So in vitro, you should be very careful with any RNA because it's easily degraded. But about the thermal stability, I think it's quite good. So it can be applied for a wide range of temperatures in vitro. So maybe not in vitro without any proteins which degrade at this temperature cells. So as the reviewer had said, if we consider some supramolecular applications for this RNA, it can be quite stable.
It will be quite stable with different molecular, for example, aptamers with thermosensor will be stable in
I think that
excuse me.
For this
Excuse me, it's not time for polemics. It's time for jurors' questions. So, shall we proceed with it? Yes. The same question was also addressed to the reviewer.
Yes, I do not know how it exactly proceed with thermal RNA, and not other sorts of RNA, because ways to protect different RNA, such as matrix RNA or other RNA or kind of cancer RNA for organism is different for every type of it. But I think thermo-RNA as mostly sensors that keepers of information sometimes in organisms should live not as long as keepers of information in organisms. It's not chromosome or such as, but in vivo, of course, in vivo, but in vitro, of course, we can keep it stable rather long time, but for this we should keep rather hard conditions. Sorry about that. Because I say it again, additions for keeping RNA in vitro are it can kill it very often, and using of it for supramolecular ways are guarded by it.
Yes, thanks. Any other questions? Yes, please.
Maybe it's a strange question, but
Your question is to.
I think to report it first, but maybe some other can
please
tell me. Does we need this kind of RNA thermometer? Maybe we should use classical metal thermometer. It's very good, really. Very accurate, 1 on 1,000 parts of Celsius degree, accurate, and we can make it quite small, micrometers. Does we really need this kind of technology?
Thanks for questions. I agree with your comments. Yes, this method is possible to use in study. Here the need очень точный temperature in cultivation cells and
Yes, please.
Of course, this RNA thermometer is not a real thermometer because melting point is just point where 50% of RNA is in our form. So it's a range of temperatures that this thermometer senses. So it's not an accurate like metal one that you said. But the point of that thermometer is that it can action after it's converted to another form, and this is the point. So you change temperature and you change some effect. And thermometer, usual thermometer is just measures the temperature, and you can use it also. You can measure the temperature and get some effect with this thermometer.
Please also review it.
Yes, RNA thermometer is not a real thermometer with hydronium or something else, but we can find another stuff to imagine it in the macro world, transistor. transistor in all the lamp TV that collapse when heat of electrochain rises. But should we use transistor when we have more and more high technology? Should we use big or awfully big FM that needed to sit it here, the whole house or the whole building, if we can use a laptop maybe or smart TV, smart watches or something else. It's rather more useful and rather more wide in its applications. I don't think this one is right, but that's all.
Thank you for your answers. Yeah. Any more questions? No. So thanks a lot. Satisfied? Yes. Please take your seats. And now we have time for jurors to put their marks.
So by the time being in these two problems we were on the subcellular and cellular level. I think we'll proceed to some other levels on the next stages. Yes, everyone ready? So let's see your marks for the reporter. For the reporter, 5, 7, 8, 6, 6, 5. For the opponent, 8, 8, 8, 8, 7, 7. And then for the review. And for the reviewer, 4, 4, 4, 3, 3, 3. That's all for this stage. Now we are going to have a break. I think we can have our time, but I offer to start exactly at 1 p.m. So we will start at 1 p.m.